So we are starting the chapter 5, that is molecular basis of inheritance. In the previous chapter you have learnt the inheritance pattern and the genetic basis of such patterns. At the time of Mendel, the nature of those factors regulating the pattern of inheritance was not clear. Over the next hundreds of years, the nature of putative genetic material was investigated, culminating in the realization that DNA, that is deoxyribonucleic acid, is the genetic material, at least for majority of organisms. In class 11th, you have learned that nucleic acids are polymers of nucleotide. Nucleic acid are polymers of nucleotide. Deoxyribonucleic acid, that is DNA, and ribonucleic acid, that is RNA, are the two types of nucleic acid found in living system. DNA acts as the genetic material in most of the organisms. RNA too, it also acts as genetic material in some viruses, mostly function as a messenger. RNA also acts as a genetic material in some viruses, mostly function as a messenger. Messenger का काम करता है mostly. RNA has an additional role as well. It functions as an adapter, structural, and in some cases as a catalytic molecule. In class 11th, you have already learned the structure of nucleotides and the way these monomer units are linked to form nucleic acid polymers. In this chapter, we are going to discuss the structure of DNA, its replication, the process of making RNA from DNA, that is transcription, the genetic code that determines the sequences of amino acids in proteins, the process of protein synthesis, that is translation, and elementary basis of their regulation. The determination of complete nucleotide sequence of human genome during last decade had set in a new era of genomics. Clear? In the last section, the essentials of human genome sequencing and its consequences will also be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is the DNA. In subsequent section, we will understand that why is the most abundant genetic material, why it is the most abundant genetic material, and what is the relationship is with the RNA. So we are studying the DNA. Let's proceed further. DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as number of nucleotides or a pair of nucleotide referred to as base pairs, present in it. This also is the characteristic of an organism. For example, a bacteriophage known as 5 into 174 has 5386 nucleotides. Bacteriophage known as 5 into 174 has 5386 nucleotides. इसको याद रख सकते हैं ऐसे 53 को प्लस किया तो आया 8, ठीक है, और 5 के बाद जो स्टार्टिंग फर्स्ट यूनिट है उसके बाद आता है 6, तो लास्ट में आएगा 6. करेक्ट आंसर इज हैज़ 5386 न्यूक्लियोटाइड्स, ऐसे याद रख सकते हो. बैक्टीरियोफेज लैम्बडा हैज़ 48502 बेस पेयर्स. बैक्टीरियोफेज लैम्बडा हैज़ 48502 बेस पेयर. इसको याद रख सकते हैं, 4 डबल होते हैं 8 और 502 याद रख सकते हैं, 48502 बेस पेयर. रट नहीं पड़ेगा वो तो, ठीक है. E. coli, that is Escherichia coli, has 4.6 into 10 to the power 6 base pair. कितने? 4.6 into 10 to the power 6 base pair. And a haploid content of human DNA is 3.3 into 10 to the power 9 base pair. Haploid content of human DNA is 3.3 into 10 to the power 9 base pair. Let us discuss the structure of such a long polymer. ठीक है, the structure of polynucleotide chain. Let us recapitulate the chemical structure of a polynucleotide chain, DNA or RNA. A nucleotide has three components: a nitrogenous base, a pentose sugar, ribose in case of RNA and deoxyribose in case of DNA, and a phosphate group. Nitrogenous base, pentose sugar and a phosphate group are the three components of nucleotide. Clear? There are two types of nitrogenous bases, that is purines. एडेनिन एंड ग्वानिन आते हैं प्यूरिन्स में, जिसे याद रख सकते हैं आग, ए से एडेनिन एंड गा से ग्वानिन. एंड pyrimidines, साइटोसिन, यूरासिल एंड थाइमिन. ये क्या है? पायरिमिडीन्स. पायरिमिडीन्स है साइटोसिन, यूरासिल एंड थाइमिन. साइटोसिन इज कॉमन फॉर बोथ डीएनए एंड आरएनए. कौन कॉमन है? साइटोसिन इज कॉमन फॉर both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA at the place of thymine. A nitrogenous base is linked to the OH of 1' pentose sugar through a N-glycosidic linkage to form a nucleoside. Nitrogenous base is linked to the OH of first carbon पेंटोस शुगर थ्रू ए एन ग्लाइकोसिडिक लिंकेज टू फॉर्म ए न्यूक्लियोसाइड. कौन से होगा? फर्स्ट कार्बन जो पेंटोस शुगर का OH उससे लिंक होगा, न्यूक्लियोसाइड बनेगा. सच एस एडिनोसिन और डीऑक्सीडेनोसाइन, डीऑक्सी एडिनोसाइन, ग्वानोसाइन और डीऑक्सी ग्वानोसिन, साइटिडिन और डीऑक्सीसाइटिडिन एंड यूरिडिन और डीऑक्सीथाइमिडिन. सब क्या है? ये न्यूक्लियोसाइड्स. व्हेन अ फॉस्फेट ग्रुप इज लिंक्ड टू OH ऑफ 5' कार्बन ऑफ ए न्यूक्लियोसाइड थ्रू फॉस्फोएस्टर लिंकेज, थ्रू फॉस्फोएस्टर लिंकेज. कौन सा? फॉस्फोएस्टर लिंकेज. करस्पॉन्डिंग न्यूक्लियोटाइड that is or deoxynucleotide depending upon the type of sugar present is formed. When a phosphate group is linked to OH of a 5' carbon of a nucleoside through phosphoester linkage, ये इंपॉर्टेंट है. कौन सा लिंकेज? फॉस्फोएस्टर लिंकेज. करस्पॉन्डिंग न्यूक्लियोटाइड इज फॉर्म्ड. टू न्यूक्लियोटाइड्स आर लिंक्ड थ्रू 3' 5' फॉस्फोडाइस्टर लिंकेज टू फॉर्म ए डाइन्यूक्लियोटाइड. टू न्यूक्लियोटाइड्स आर लिंक्ड थ्रू 3' 5' phosphodiester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form polynucleotide chain. A polymer thus formed has at one end a free phosphate moiety at the 5' end of a sugar, which is referred to as 5' end of the polynucleotide chain. Similarly, at the other end of the polymer, the sugar has a free OH of 3' carbon group, which is referred to as 3' end of the polynucleotide chain. Similarly, at the other end of the polymer, the sugar has a free OH of 3' carbon group, which is referred to as 3' end of the polynucleotide chain. The backbone of a polynucleotide chain is formed due to sugar and phosphate. बैकबोन किससे फॉर्म होती है? शुगर एंड फॉस्फेट से. द बैकबोन ऑफ अ पॉलीन्यूक्लियोटाइड चेन इज फॉर्म्ड ड्यू टू शुगर एंड अ फॉस्फेट. द नाइट्रोजनस बेसेस लिंक्ड टू a sugar moiety project from the backbone. The nitrogenous bases linked to sugar moiety project from the backbone. In RNA, every nucleotide residue has an additional OH group present at 2' position in the ribose. Also, in RNA, the uracil is found at the place of thymine. Also, in RNA, the uracil is found at the place of thymine, जिसे कहते हैं 5-methyl uracil, another chemical name of thymine. Thymine का another chemical name है 5-methyl uracil. Clear? DNA as an acidic substance present in nucleus was first identified by Friedrich Miescher in 1869. DNA as an acidic substance present in nucleus was first identified by Friedrich Miescher in 1869. He named it as a nuclein. However, due to technical limitation in isolating such a long polymer intake, the elucidation of the structure of DNA remained elusive for a very long period of time. It was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but very famous double helix model for the structure of DNA. One of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. However, this proposition was also based on the observation of Erwin Chargaff that for a double-stranded DNA, the ratio between adenine and thymine and guanosine and cytosine are constant and equals 1. Ratio between adenine and thymine means AT and guanosine and cytosine, जिसे याद रख सकते हैं AT और GC are constant. The base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore, if the sequences of base in one strand is known, then the sequences in other strand can be predicted. Also, if each strand from the DNA, let us call it as a parental DNA, act as a template for synthesis of a new strand, the two double-stranded DNA, let us call them as a daughter DNA, thus produced would be identical to the parent DNA molecule. क्या होगी? Would be identical. Because of this, the genetic implications of the structure of DNA become very clear. The salient feature of the double helix structure of DNA are as follows. Okay? So we are reading the salient features of double helix structure of DNA. So the first point is, it is made up of two polynucleotide chain, where the backbone is constituted by the sugar phosphate and the bases project inside. Bases project inside. Change करके outside कर देगा, but correct है inside. First point is, it is made up of two polynucleotide chains where the backbone is constituted by sugar phosphate and the bases project inside. Second point is, the two chains have anti-parallel polarity. It means if the one chain has the polarity 5' to 3', the other is 3' to 5'. Third point is, the bases in two strands are paired through hydrogen bond. किससे? Hydrogen bond, forming base pairs. Adenine forms two hydrogen bond with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bonds. As a result, always a purine comes opposite to pyrimidine. This generates approximately uniform distance between the two strands of the helix. Fourth point is, the two chains are coiled in right-handed fashion. The pitch of the helix is 3.4 nanometer. A nanometer is 1 billionth of the meter, that is 10 to the power minus 9 meter. A nanometer is 1 billionth of a meter, that is 10 to the power minus 9 meter. Again I am repeating it. The two chains are coiled in right-handed fashion. The pitch of the helix is 3.4 nanometer. Pitch of helix is 3.4 nanometer. And there are roughly 10 base pair in each turn. And there are roughly 10 base pair in each turn. Consequently, the distance between a base pair in a helix is approximately 0.34 nanometer. Consequently, the distance between a base pair in a helix is approximately 0.34 nanometer. Again I am repeating it. Consequently, the distance between a base pair in a helix is approximately 0.34 nanometer. So the fifth point is the plane of one base pair takes over the other in double helix. This, in addition to hydrogen bonds, confers stability of the helical structure. Clear? The plane of one base pair takes over the other in double helix. This, in addition to hydrogen bonds, confers stability of the helical structure. Clear hai? Compare the structure of purines and pyrimidines. Can you find out why the distance between the two polynucleotide chains in DNA remains almost constant? Can you find out why the distance between two polynucleotide chains in DNA remains almost constant? The preposition of a double helix structure for DNA and its simplicity in explaining the genetic implication become revolutionary. Very soon Francis Crick proposed the central dogma in a molecular biology, which states that the genetic information flows from DNA to RNA and RNA to protein. यहाँ पर डायग्राम भी दिया हुआ है सेंट्रल डॉगमा का, जिसमें रेप्लिकेशन दिया हुआ है डीएनए, ठीक है, डीएनए से डीएनए, ठीक है, डीएनए से एमआरएनए ट्रांसक्रिप्शन, एमआरएनए से प्रोटीन कहते हैं ट्रांसलेशन, ठीक है. In some viruses, the flow of information is in reverse direction, that is from RNA to DNA. Can you suggest a simple name to this process? That is reverse transcription. Simple. So next is packaging of DNA helix. Taken the distance between two consecutive base pair as 0.34 nanometer, that is 0.34 into 10 to the power minus 9 meter. If the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pair with the distance between two consecutive base pair, that is 6.6 into 10 to the power 9 base pair into 0.34 into 10 to the power minus 9 meter per base pair, it comes out to be approximately 2.2 meter. कितनी आएगी? approximately 2.2 meters. A length that is far greater than the dimension of a typical nucleus, that is approximately 10 to the power minus 6 meter. How is such a long polymer packaged in a cell? कैसे पैक करते हैं इसे? If the length of E. coli is 1.36 millimeter, can you calculate the number of base pairs in E. coli? Question सुनो वापस. If the length of E. coli DNA is 1.36 mm, can you calculate the number of base pairs in E. coli? Yes, we can calculate it. The number of base pair in the E. coli formula लगा के total number of base pair is equal to total length of DNA upon distance between two base pair. ठीक है, उसका आंसर आएगा approximately 4 into 10 to the power 6 base pair. ठीक है, that is the answer of that. In a prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. DNA, being negatively charged, is held with some proteins that have positive charges in a ring termed as nucleoid. The DNA in nucleoid is organized in large loops held by proteins. The DNA in nucleoid is organized in large loops held by proteins. Clear? In eukaryotes, this organization is much more complex. There is a set of positively charged basic proteins called histones. A protein acquires charge depending upon the abundance of amino acids residues with the charged side chains. Histones are rich in the basic amino acids such as lysine and arginine. इंपॉर्टेंट लाइन है, बहुत बार क्वेश्चन पूछा गया है, बहुत सारे मॉक टेस्ट में मिल भी जाएगा. Histones are rich in the basic amino acids such as lysine and arginine. Both the amino acid residues carry positive charge in their side chains. Histones are organized to form a unit of 8 molecules called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. A typical nucleosome contains 200 base pair of DNA helix. Nucleosomes constitute the repeating unit of a structure in nucleus called chromatin. Thread-like stained colored body seen in nucleus. Nucleosomes constitute the repeating unit of a structure in nucleus called chromatin. Nucleosome constitute the repeating unit of a structure in nucleus called chromatin. Thread-like stained color bodies seen in nucleus. The nucleosomes in chromatin are seen as beads on strings, a structure when viewed under electron microscope. कौन से microscope? When viewed under electron microscope. Theoretically, how many such beads, that is nucleosomes, do you imagine are present in a mammalian cell? The beads on string structure in chromatin is packaged to form chromatin fiber that are further coiled and condensed at metaphase stage of cell division to form chromosome. कौन सी स्टेज पे? Metaphase stage पे of cell division to form chromosomes. The packaging of chromatin at higher level requires additional set of protein that collectively referred to as non-histone chromosomal proteins, that is NHC. Again I am repeating that line before wali. The beads on string structure in chromatin is packaged to form chromatin fiber that are further coiled and condensed at metaphase stage of cell division to form chromosome. The packaging of chromatin at a higher level requires additional set of proteins that collectively are referred to as non-histone chromosomal proteins, NHC. In a typical nucleus, some ring of chromatin are loosely packed and stains light, while and are referred to as euchromatin. In a typical nucleus, some rings of chromatin are loosely packed and stains light and are referred to as euchromatin. The chromatin that is more densely packed and stains dark are called heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. So I am एक्सप्लेनिंग दिस इन समरी. यू क्रोमेटिन होते हैं वो लूजली पैक्ड होते हैं जिन्हें लाइटली स्टेन कहते हैं और वो ट्रांसक्रिप्शनली एक्टिव होते हैं. हेट्रोक्रोमेटिन होते हैं डेंसली पैक्ड होते हैं और डार्क स्टेन होते हैं, और हेट्रोक्रोमेटिन जो होते हैं इनएक्टिव होते हैं. क्लियर? नेक्स्ट इज द सर्च फॉर जेनेटिक मटेरियल. Even though the discovery of nuclein by Mescher and the proposition for the principle of inheritance by Mendel were almost at the same time, but that the DNA act as genetic material took long time to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor John Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search of the chromosome located in the nucleus of most cells. But the question of what molecule was actually the genetic material had not been answered. Next, we are going to read transforming principle. In 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumoniae, a bacterium responsible for pneumonia, witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism, that is bacteria, had changed in physical form. When Streptococcus pneumoniae, that is pneumonococcus, bacteria are grown on culture plate. Some produce smooth, shiny colonies, while other produces rough colonies. शाइनी कॉलोनीज को कैपिटल एस से रिप्रेजेंट करेंगे और रफ कॉलोनीज को कैपिटल आर से रिप्रेजेंट करेंगे. दिस इज बिकॉज द एस स्ट्रेन बैक्टीरिया हैव अ म्यूकस पॉलीसेकेराइड कोट while the R strain does not. S strain के पास है म्यूकस पॉलीसेकेराइड कोट, ठीक है, while R does not है. Mice infected with the S strain virulent die from pneumonia infection, but mice infected with the R strain do not develop pneumonia. S का इंजेक्शन लगा दिया तो चूहा क्या हो गया, मर गया, ठीक है, और आर का स्ट्रेन का लगाया तो चूहा जिंदा है, उसमें कुछ नहीं हुआ, उसमें निमोनिया नहीं हुआ, ठीक है. यहाँ पे दिया हुआ है चार्ट में, एस स्ट्रेन इंजेक्ट इनटू माइस, माइस डाई, आर स्ट्रेन इंजेक्ट इनटू माइस, माइस अलाइव, चूहा जिंदा है. ग्रिफ्थ वाज एबल टू किल बैक्टीरिया बाय हीटिंग दैम. ग्रिफ्थ वाज एबल टू किल बैक्टीरिया बाय हीटिंग दैम. ही ऑब्जर्व्ड दैट heat killed S strain bacteria injected into mice did not kill them. When he injected a mixture of heat killed S and live R bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice. हो गया खेला. He observed that heat killed S strain bacteria injected into mice did not kill them. When he injected a mixture of heat-killed S and live R bacteria, the mice died. Moreover, he recovered living S bacteria from dead mice. चार्ट में दिया हुआ है S strain heat killed inject into mice, mice live. जब S strain heat killed किया और उसे R strain के साथ इंजेक्ट किया माइस में तो माइस मर गया, मतलब चूहा मर गया. He concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. किससे? heat killed जो S strain bacteria उससे क्या हो गया, somehow transfer हो गया R strain bacteria. Some transforming principle transferred from the heat killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of the genetic material. However, the biochemical nature of genetic material was not defined from his experiment. ठीक है, गई भैंस पानी. So the next experiment is biochemical characterization of transforming principle. Prior to the work of Oswald Avery, Colin MacLeod and Maclean McCarty in 1933 to 44, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principles in Griffith experiment. They purified biochemicals, that is proteins, DNA, RNA, etc., from the heat-killed S cells to see which ones could transform live R cells into the S cell. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. क्या उन्होंने डिस्कवर किया, डीएनए अकेला एस बैक्टीरिया को क्या कर दिया उसने, एस बैक्टीरिया ने क्या कर दिया, आर बैक्टीरिया टू बिकम ट्रांसफॉर्म, ठीक है. दे ऑल्सो डिस्कवर्ड दैट प्रोटीन डाइजेस्टिंग एंजाइम्स, दैट इज प्रोटीएसेस, and RNA digesting enzyme, that is RNase, did not affect the transformation. So the transforming substance was not a protein or RNA. Digestion with DNase did inhibit transformation, suggesting that the DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced. Can you think of any difference between DNase and DNase? Can you think? First of all, I am repeating that line once again. They also discovered that protein digesting enzyme, that is proteases, and RNA digesting enzyme RNase did not affect transformation. So the transforming substance was not a protein or RNA. Digestion with DNase did inhibit transformation, suggesting that the DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced. Can you think of any difference between DNase and DNase? DNA is the nucleic acid, and while DNase is an enzyme. Clear? Let's proceed further. So the next topic is the genetic material is DNA. The unequivocal proof that DNA is the genetic material come from the experiment of Alfred Hershey and Martha Chase in 1952. In the year 1952, Alfred Hershey and Martha Chase, they worked with virus that infect bacteria called bacteriophages. They worked with virus that infect bacteria called bacteriophages. The bacteriophage attaches to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particles. Hershey and Chase worked to discover whether it was protein or DNA from the viruses that enter the bacteria. They grew some virus on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA but not radioactive protein, because DNA contains phosphorus but protein does not. Clear? Similarly, viruses grown on radioactive sulfur contained radioactive protein but not radioactive DNA, because DNA does not contain sulfur. DNA does not contain sulfur. Clear? Radioactive phages were allowed to attach to the E. coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. The virus particles were separated from the bacteria by spinning them in a centrifuge. Clear? Spinning them in a centrifuge. Bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with the virus that had radioactive protein were not radioactive. This indicates that protein did not enter the bacteria from the virus. DNA is therefore the genetic material that is passed from virus to bacteria. Clear? डायग्राम दिया हुआ है यहाँ पे हर्शी एंड चेस एक्सपेरिमेंट का, जिसमें ऊपर बैक्टीरियोफेज दिया हुआ है 35S लेबल्ड प्रोटीन कैप्सूल से और रेडियोएक्टिव 32P लेबल्ड डीएनए से। और फर्स्ट स्टेप है इन्फेक्शन, फिर ब्लेंडिंग, फिर सेंट्रीफ्यूगेशन। ठीक है, जो फर्स्ट डायग्राम है उसमें रेडियो एक्टिव, जिसमें प्रोटीन कैप्सूल है, उसमें नो रेडियो एक्टिव 35 सेल्स डिटेक्टेड इन सेल प्लस रेडियो एक्टिव 35S डिटेक्टेड इन सुपरनेट्स। सेकंड जो है उसमें रेडियो एक्टिव 32P डिटेक्टेड इन सेल, नो रेडियो एक्टिव डिटेक्टेड इन सुपरनेट। सो द नेक्स्ट इज द प्रॉपर्टीज ऑफ जेनेटिक मटेरियल DNA versus RNA. From the foregoing discussion, it is clear that the debate between protein versus DNA as the genetic material was unequivocally resolved from the Hershey-Chase experiment. It became an established fact that it is a DNA that acts as genetic material. However, it's subsequently become clear that in some viruses, RNA is the genetic material. However, it subsequently become clear that in some viruses, RNA is the genetic material. For example, tobacco mosaic virus, QB bacteriophage, etc. इनमें कौन है RNA genetic material? किसमें? टोबैको मोसेक वायरस में और क्यूबी बैक्टीरियोफेज में आरएनए जेनेटिक मटेरियल होता है। क्लियर? आंसर टू सम ऑफ द क्वेश्चन सच एज व्हाई डीएनए इज द प्रीडोमिनेंट जेनेटिक मटेरियल whereas RNA performs dynamic functions of messenger, and adapter has to be found from the differences between chemical structure of the two nucleic acid molecule. Clear? Can you recall the two chemical differences between DNA and RNA? A molecule that can act as a genetic material must fulfill the following criteria. First, it should be able to generate its replica, that is replication. Second point, it should be stable chemically and structurally. First है, it should be able to generate its replica, that is replication. Second is, it should be stable chemically and structurally. Chemically और structurally क्या होना चाहिए? Stable. Third है, it should provide the scope for slow change, that is mutation, that are required for evolution. It should provide the scope for slow changes, that is mutations, that are required for evolution. Fourth point is, it should be able to express itself in the form of Mendelian characters. If one examines each experiment one by one, because of rule of best pairing and complementary, both the nucleic acid, that is DNA and RNA, have the ability to generate their duplication. Both DNA and RNA have the ability to direct their duplications. The other molecules in the living system, such as protein, fails to fulfill first criteria itself. The genetic material should be stable enough not to change with different stages of life cycle, age, or with change in physiology of the organism. ठीक है, क्या होना चाहिए? स्टेबल इनफ होना चाहिए, चेंज नहीं होना चाहिए लाइफ साइकिल, एज या फिर चेंज इन फिजियोलॉजी के साथ. ठीक है. Stability as one of the property of genetic material was very evident in Griffith's transforming principle itself. That heat which killed the bacteria at least did not destroy some of the properties of genetic material. Which killed the bacteria at least did not destroy some of the properties of genetic material. This now can easily be explained in light of the DNA. That the two strains, being complementary, if separated by heating, come together when appropriate conditions are provided. Clear? Further, second dash OH group present at every nucleotide in RNA is a reactive group and makes RNA labile and easily degradable. Further, second dash OH group present at every nucleotide in RNA is a reactive group and makes RNA labile and easily degradable. RNA is also now known to be catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. DNA chemically is less reactive and structurally more stable when compared to RNA. Clear? Therefore, among the two nucleic acids, the DNA is the better genetic material. कौन है better genetic material? DNA. In fact, the presence of thymine at the place of uracil also confers additional stability to DNA. Detailed discussion about this requires understanding of the process of repair in DNA, and you will study this process in higher classes. Both DNA and RNA are able to mutate. In fact, RNA being unstable mutate at a rate faster. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. जिस वायरस का RNA जीनोम शॉर्टर लाइफ स्पेन का होता है वो क्या होते हैं? म्यूटेट करते हैं और क्या होते हैं, एवॉल्व होते हैं फास्टर. शॉर्टर लाइफ स्पेन, म्यूटेट एंड एवॉल्व फास्टर. RNA can directly code for the synthesis of proteins, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of protein. The protein synthesizing machinery has evolved around RNA. The above discussion indicate that both DNA and RNA can function as genetic material, but DNA being more stable is preferred for storage of genetic information. For the transformation of the genetic information, RNA is better. स्टोरेज के लिए कौन है, DNA बेटर है. ट्रांसफॉर्मेशन के लिए कौन है, RNA बेटर है. क्लियर, ठीक है. सो नेक्स्ट इज RNA world. We are going to read the RNA world. From foregoing discussion, an immediate questions become evident, which is the first genetic material. It shall be discussed in detail in the chapter on chemical evolution. But briefly, we shall highlight some of the facts and points. RNA was the first genetic material. There is now enough evidence to suggest that essential life process such as metabolism, translation, splicing, etc. evolved around RNA. RNA used to act as a genetic material as well as catalyst. There are some important biochemical reactions in living system that are catalyzed by RNA catalyst and not by protein enzyme. Are catalyzed by RNA catalyst and not by protein enzymes. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA is evolved from RNA. Therefore, DNA has evolved from RNA with chemical modifications that make it more stable. DNA being double-stranded and having complementary strain further resists changes by evolving a process of repair. Next is replication. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, that is as follows: "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible coping mechanism for the genetic material." Watson and Crick in 1953. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible coping mechanism for the genetic material. The scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. The scheme suggested that the two strands would separate and act as template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. The scheme was termed as semi-conservative DNA replication. Next, the experimental proof. It is now proven that DNA replicates semi-conservatively. It was shown first in Escherichia coli. It was first shown in E. coli and subsequently in higher organisms such as plants and human cells. Matthew Meselson and Franklin Stahel performed the following experiment in 1958. Matthew Meselson and Franklin Stahel performed the following experiment in 1958. First point is, they grew E. coli in a medium containing 15NH4Cl. 15N is the heavy isotope of nitrogen, as the only nitrogen source for many generations. Again I am repeating it. They grew equally in a medium containing 15 NH4Cl. 15N is the heavy isotope of nitrogen, as the only nitrogen source for many generations. The result was that 15N was incorporated into newly synthesized DNA, as well as the nitrogen containing compound. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride, that is CaCl, density gradient. Please note that 15N is not a radioactive isotope and it can be separated from 14N only based on densities. Clear? The heavy DNA molecule could be distinguished from the normal DNA by the centrifugation in a cesium chloride, that is CaCl, density gradient. Second point is, then they transferred the cells into the medium with normal 14NH4Cl and took samples at various definite time intervals as the cells multiplied and extracted the DNA that remained as double-stranded helices. The various samples were separated independently on CaCl gradient, that is cesium chloride gradients, to measure the densities of DNA. Can you recall what centrifugal force is and think why a molecule with higher mass density would sediment faster? Again, I am repeating it. Can you recall what centrifugal force is? What centrifugal force is? And think why a molecule with a higher mass density would sediment faster. जिसका मास हायर होगा वो सेडिमेंट फास्टर क्यों करता है? So the centrifugal force is a force that acts away from the center of a rotating object. It is a pseudo force. ठीक है? अब what is the explanation of it? It is asked that why a molecule with a higher mass density would sediment faster. A molecule with higher mass or density sediments faster because of the greater centrifugal force acting on it. This is the answer of it. Because what? Repeat it again. Because what is acting on it? Because greater centrifugal force is because of the greater centrifugal force acting on it. Clear? अब यहाँ पर रिजल्ट दिया हुआ है डायग्राम में मैसेल्सन एंड स्टेल्सन का एक्सपेरिमेंट, जिसमें 15N DNA दिया हुआ है। 20N के बाद 15N DNA 14N DNA बन गया फर्स्ट जनरेशन में, फिर उनको अलग कर दिया वापस 14N DNA 14N DNA 14N अलग कर दिया, 20 मिनट 40 मिनट लाइट हाइब्रिड हैवी। ठीक है, डायग्राम दिया हुआ है। सो द थर्ड पॉइंट इज थस the DNA that was extracted from the culture one generation after the transfer from 15N to 14N medium, that is after 20 minutes, equally divides in 20 minutes. कितने मिनट में डिवाइड होता है इक्वली? Equally divides in 20 minutes. Had a hybrid or intermediate density. DNA extracted from the culture after another generation, that is after 40 minutes, that is second generation. DNA extracted from the culture after another generation, that is after 40 minutes, that is second generation, was composed of equal amounts of this hybrid DNA and of light DNA. If E. coli was allowed to grow for 80 minutes, if E. coli was allowed to grow for 80 minutes, then what would be the proportions of light and hybrid density DNA molecule? बताओ, आंसर करो। If the E. coli is allowed to grow for 80 minutes, the proportion of light and hybrid density molecule will be 87.5% light and 12.5% hybrid. This is because E. coli divides every 20 minutes, so there are 4 generations after 80 minutes. ठीक है, फर्स्ट जनरेशन में ऑल स्ट्रैंड हाइब्रिड होंगे, सेकंड में 50% ऑफ DNA इज लाइट एंड 50% इज हाइब्रिड, थर्ड में 25% इज हाइब्रिड एंड 75% इज लाइट, और फोर्थ जनरेशन में 12.5% हाइब्रिड होंगे और 87.5% लाइट होंगे। क्लियर? Very similar experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome was performed on Vizia faba, that is faba beans, by Taylor and colleagues in 1958. In 1958, the experiment proved that the DNA in chromosome also replicate semi-conservatively. The DNA in chromosome also replicate semi-conservatively. Next topic is the machinery and the enzymes. In living cells such as E. coli, the process of replication requires a set of catalyst enzymes. The main enzyme is referred to as DNA-dependent DNA polymerase. The main enzyme referred to as DNA-dependent DNA polymerase. Since it uses a DNA template to catalyze the polymerization of deoxynucleotides, these enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. The main enzyme is referred to as DNA-dependent DNA polymerase, since it uses a DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. E. coli that has only 4.6 into 10 to the power 6 base pair. E. coli that has only 4.6 into 10 to the power 6 base pair. Compare it with the human whose diploid content is 6.6 into 10 to the power 9 base pair. Completes the process of replication within 18 minutes. कितने मिनट में करता है? 18 minutes में करता है E. coli. That means the average rate of polymerization has to be approximately 2000 base pair per second. पर सेकंड की जगह मिनट कुछ भी कर सकता है, बट करेक्ट है 2000 base pair per second. Again I am repeating that the E. coli that has only 4.6 into 10 to the power 6 base pair completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 base pair per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy. Any mistake during replication would result into mutations. Furthermore, energetically, replication is very expensive process. Deoxyribonucleoside triphosphate serves dual purposes. Deoxyribonucleoside triphosphates serve dual purposes. In addition to acting as substrates, they provide energy for polymerization reaction. The two terminal phosphates in a deoxynucleoside triphosphate are high energy phosphates, same as in case of ATP. In addition to acting as substrate, they provide energy for polymerization reaction. The two terminal phosphates in a deoxynucleoside triphosphate are high energy phosphates, same as in case of ATP. In addition to DNA-dependent DNA polymerases, many additional enzymes are required to complete the process of replication with high degree of accuracy. In addition to DNA-dependent DNA polymerases, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length, that is due to very high energy requirement, The replication occurs within a small opening of the DNA helix referred to as replication fork. The DNA-dependent DNA polymerase catalyzes polymerization only in one direction, that is 5' to 3'. Polymerizes only in one direction, that is 5' to 3'. This creates some additional complications at the replicating fork. This creates some additional complications at the replicating fork. Consequently, on one strain, the template with polarity 3' to 5', the replication is continuous. कौन सी पे है continuous? 3' to 5'. 3' to 5'. The replication is continuous. While on the other, the template with polarity 5' to 3', it is discontinuous. जो 5', 3' की होती है वो होती है discontinuous, और 3' से 5' वाली होती है replication is continuous. ठीक है. Discontinuously जिसमें 5' से 3' polarity होती है, the discontinuously synthesized fragments are later joined by the enzyme DNA ligase. The DNA polymerases on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. There is a definite line in E. coli DNA where the replication originates. Such lines are termed as origin of replication. It is because of the requirement of the origin of replication that a piece of DNA needed to be propagated during recombinant DNA procedures requires a vector. The vectors provide the origin of application. कौन provide करते हैं? The vectors provide the origin of replication. Clear? Further, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place at a phase of the cell cycle. The replication of DNA and cell division should be highly coordinated. The replication of DNA and cell division cycle should be highly coordinated. A failure in cell division after DNA replication results in polyploidy, a chromosomal anomaly. A failure in cell division after DNA replication. A failure in cell division after DNA replication results in a polyploidy, that is chromosomal anomaly. You will learn the detailed nature of origin. You will learn the detailed nature of origin and the process occurring at this site in a higher class. डायग्राम दिया हुआ है रेप्लिकेटिंग फोक का, जिसमें कंटिन्यूअस है 3 days से 5 days वाली चेन और डिसकंटिन्यूअस है 5 days से 3 days वाली. ठीक है. नेक्स्ट टॉपिक इज ट्रांसक्रिप्शन. उससे पहले आपको मैं एक सॉन्ग सुना देता हूँ. ठीक है. कौन सा सुनाता हूँ? साहे अखिया चुरा ले पर्दे से ओ बीबा मैनू दूर ना करे, पास आया हूँ बड़ी मुश्किल से ओ बीबा मैनू दूर ना करे. इनफ. सुन लिया? चलो, द नेक्स्ट टॉपिक इज ट्रांसक्रिप्शन. द प्रोसेस ऑफ कॉपिंग जेनेटिक इंफॉर्मेशन from one strain of the DNA into RNA is termed as transcription. The process of copying genetic information from one strain of the DNA into RNA is termed as transcription. Here, also the principle of complementary governs the process of transcription, except the adenosine complements now forms base pair with uracil instead of thymine. Except the adenosine complements now forms base pair with the uracil instead of thymine. Clear? So the next is However, unlike in the process of replication, which once set in, the total DNA of an organism gets duplicated. In transcription, only a segment of DNA and only one of the strands is copied into RNA. In transcription, only a segment of DNA and only one of the strands is copied into RNA. This necessitates defining the boundaries that would demarcate the line and the strand of DNA that would be transcribed. That would demarcate the line and the strand of DNA that would be transcribed. Why both the strands are not copied during transcription? Has the simple answer. First, if both the strands act as a template, they would code for RNA molecule with different sequences. Remember, complementary does not mean identical. And in turn, if they code for protein, the sequence of amino acid in the proteins would be different. First, if both strands act as a template, they would code for RNA molecule with different sequences. And in turn, if they code for protein, the sequence of amino acid in the protein would be different. Hence, one segment of DNA would be coding for two different proteins. One segment of DNA would be coding for two different proteins. And this would complicate the genetic information transfer machinery. Second, the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein, and the exercise of transcription would become a futile. Second, the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein, and the exercise of transcription would become futile one. Clear? So the next is the transcription unit. We are going to read the topic transcription unit. A transcription unit in DNA is defined primarily by the 3 lines in the DNA. कितने lines? 3 lines. First line is promoter, second is structural gene, third is terminator. First is a promoter, second is the structural gene, third is a terminator. समझ आ गया? हो गया माइंड में सेट? क्लियर? चलो आगे बढ़ते हैं. Let's proceed further. There is a convention in defining the 2 strands of DNA in the structural gene of the transcription unit. There is a convention in defining the 2 strands of the DNA, the structural gene of a transcription unit. Since the 2 strands have opposite polarity and the DNA-dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5' to 3'. RNA, that is DNA-dependent RNA polymerase, also catalyze polymerization in only one direction, that is 5' to 3'. The strand that has the polarity 3' to 5' act as template. इसे ऐसे याद रख सकते हो 3D से. 3' वाली जो होती है वो टेम्पलेट होती है, 3' से 5' वाली 3D, and is also referred to as template strand. The other strand, which has the polarity 5' to 3' and the sequence same as RNA except thymine at the place of uracil, is displaced during transcription. Strangely, this strand which does not code for anything is referred to as coding strand. This strand which does not code for anything is referred to as coding strand. किसी के लिए कोड ही नहीं कर रही है बट फिर भी कॉडिंग स्ट्रैंड कहते हैं इसको. ऑल द रेफरेंस पॉइंट वाइल डिफाइनिंग ए ट्रांसक्रिप्शन यूनिट इज मेड विद कोडिंग स्ट्रैंड. जब कुछ कर ही नहीं है तो बस वो एक ही काम करेगी, रेफरेंस पॉइंट. ठीक है. ऑल द रेफरेंस पॉइंट वाइल डिफाइनिंग ए transcription unit is made with the coding string. To explain the point, a hypothetical sequence from the transcription unit is represented below, in which 3 days to 5 prime की चीज को टेम्पलेट स्ट्रेन कहते हैं और 5 प्राइम से 3 प्राइम को कोडिंग स्ट्रेन कहते हैं. Can you now write the sequence of RNA transcribed from the above DNA? You can write that. Clear? So the next is systematic structure of a transcription unit. तो 3 days की अपर प्राइम पे होता है प्रमोटर होता है और टर्मिनेटर होता है 5 प्राइम पे फर्स्ट वाले की, ठीक है. और जो डाउन स्ट्रीम होती है उसे कहते हैं कोडिंग स्ट्रीम. ऊपर वाले को टेम्पलेट स्ट्रक्चरल जीन दिया हुआ है. प्रमोटर जो होता है 5 days डाउन प्राइम पे होता है, ठीक है. द प्रमोटर एंड द टर्मिनेटर फ्लैंक द स्ट्रक्चरल जीन इन ए ट्रांसक्रिप्शन यूनिट. The promoter is said to be located towards 5 dash end, that is upstream of the structural gene. The reference is made with respect to the polarity of coding string. Again I am repeating that the promoter and the terminator flank the structural gene in a transcription unit. The promoter is said to be located towards the 5 dash end, that is upstream of the structural gene. That the reference is made with respect to the polarity of coding string. It is a DNA sequence that provides binding site for RNA polymerase, and it is the presence of a promoter in the transcription unit that also defines the template in the coding string. And it is the presence of promoter in a transcription unit that also defines the template in the coding strings. By switching its position with terminator, the definition of coding and the template string could be reversed. The terminator is located towards 3 dash end, that is downstream of the coding string. Terminator is located towards 3 dash end, that is downstream of the coding string, and जो promoter होता है is located towards 5 dash end, that is upstream of the structural gene, ठीक है. Terminator is located toward 3 dash end downstream of the coding string, and it usually defines the end of the process of transcription. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with regulation of gene expression. Next is transcription unit and the gene. A gene is defined as the functional unit of inheritance. A gene is defined as the functional unit of inheritance. Though there is no ambiguity that the genes are located on the DNA, it is difficult to literally define a gene in terms of DNA sequence. The DNA sequence coding for tRNA or rRNA molecule also define a gene. However, by defining a cistron as a segment of DNA coding for polypeptide, ठीक है, cistron as a segment of DNA coding for polypeptide, the structural gene in a transcription unit could be said as monocistronic, mostly in eukaryotes, or polycistronic, mostly in bacteria or prokaryotes. ऐसे याद रख सकते हैं. पॉलीसिस्ट्रॉनिक मीन्स जिसका स्टार्टिंग पी से है वो प्रोकैरियोट्स में होते हैं, ठीक है. मोनोसिस्ट्रॉनिक यूकैरियोट्स में होते हैं. इन यूकैरियोट्स द मोनोसिस्ट्रॉनिक structural gene have interrupted coding sequences. The monocistronic structural gene have interrupted coding sequences. The genes in eukaryotes are split. The coding sequences or expressed sequence are defined as exons. The coding sequences or expressed sequences are defined as exons. Exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by introns. Introns or intervening sequences do not appear in mature or processed RNA. Introns or intervening sequences do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of a gene in terms of DNA segment. Inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as regulatory gene, even though this sequence do not code for any RNA or protein. Again, I am repeating that inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as regulatory genes, even though this sequence do not code for any RNA or protein. Clear? Next is types of RNA and the process of transcription. In bacteria, there are three major types of RNAs, that is mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. All three RNAs are needed to synthesize a protein in a cell. The mRNA provides the template. tRNA brings amino acids and reads the genetic code, and RNA and rRNAs plays structural and catalytic role during translation. ठीक है, अगेन आई एम रिपीटिंग दैट mRNA क्या प्रोवाइड करता है? टेम्पलेट. tRNA brings amino acids and reads the genetic code. tRNA amino acids को bring लेकर आता है, ठीक है, और क्या करता है? जेनेटिक कोड को रीड करता है. rRNA क्या करता है? प्ले स्ट्रक्चरल एंड कैटालाइटिक रोल ड्यूरिंग ट्रांसलेशन. चेंज करके ट्रांसक्रिप्शन कर सकते हैं यहाँ पे, बट करेक्ट है ट्रांसलेशन. ठीक है, क्वेश्चन भी बनाते जा रहा हूँ. rRNA play structural and catalytic role during translation. There is single DNA dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria. There is single DNA dependent RNA polymerase that catalyzes transcription of all types of RNA in a bacteria. RNA polymerase that catalyzes transcription of all types of RNA in bacteria. ठीक है, there is a single DNA dependent RNA polymerase that catalyzes transcription of all types of RNA in a bacteria. RNA polymerase binds to a promoter and initiates transcription, that is initiation. It uses nucleoside triphosphate as substrate and polymerizes in a template dependent fashion following the rule of complementarity. Following the rule of complementarity. डायग्राम दिया हुआ है प्रोसेस ऑफ ट्रांसक्रिप्शन इन बैक्टीरिया, जिसमें इनिशिएशन दिया हुआ है, फिर एलोंगेशन, फिर टर्मिनेशन. ठीक है, सिग्मा फैक्टर दिया हुआ है प्रमोटर की साइड और रॉ फैक्टर दिया हुआ है टर्मिनेशन की साइड. ठीक है, टर्मिनेट करने के लिए. ठीक है, सो द नेक्स्ट इज इट समहाउ आल्सो फैसिलिटेट्स opening of the helix and continuous elongation. Only a short stage of RNA remains bound to the enzyme. Once the polymerase reaches the terminator rime, the nascent RNA falls off, so also the RNA polymerase. This results in termination of transcription. Clear? Once again I am repeating that once the polymerase reaches the terminator rime, the nascent RNA falls off, so also the RNA polymerase. This results in termination of transcription. An intriguing question is that how is the RNA polymerase able to catalyze all the three steps which are initiation, elongation and termination? The RNA polymerase is only capable of catalyzing the process of elongation. RNA polymerase polymerase is only capable of catalyzing the process of elongation. किसके capable है? बस elongation के लिए. It associates transiently with the initiation factor sigma and termination factor rho to initiate and terminate the transcription. Associate transiently with the initiation factor sigma and termination factor rho to initiate and terminate the transcription, respectively. Association with this factor alter the specificity of the RNA polymerase to either initiate or terminate. In bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation takes place in the same compartment, there is no separation of cytosol and nucleus in bacteria. Many times, the translation can begin much more before the mRNA is fully transcribed. Many times, the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. Translation and transcription can be coupled in bacteria. Clear? In eukaryotes, there are two additional complexities. First, there are at least 3 RNA polymerases in the nucleus. In addition to the RNA polymerase found in the organelles, there is clear-cut division of labor. There are at least 3 RNA polymerases in the nucleus in addition to the RNA polymerase found in the organelles. There is a clear-cut division of labor. The RNA polymerase first transcribes rRNAs. The RNA polymerase first transcribes rRNA, that is 28S, 18S and 5.8S. 28S, 18S and 5.8S in RNA polymerase first. ठीक है? Whereas the RNA polymerase second transcribes precursor of mRNA, the heterogeneous nuclear RNA, that is hnRNA. Clear? Whereas the RNA polymerase third is responsible for transcription of tRNA, 5S RNA and snRNA, जिसे small nuclear RNAs कहते हैं. ठीक है? अगेन आई एम रिपीटिंग दैट RNA polymerase फर्स्ट किसका करता है ट्रांसक्राइब? rRNAs, that is 28S, 18S and 5.S. RNA polymerase second transcribe precursor of mRNA, the heterogeneous nuclear RNA, that is hnRNA. Whereas RNA polymerase third is responsible for transcription of tRNA, 5S RNA and snRNA. snRNA का full form है small nuclear RNAs. Second point is, the second complexity is that the primary transcript contains both the exon and the introns and are non-functional. Hence, it is subjected to a process called splicing, where the introns are removed and exons are joined in a defined order. hnRNA undergoes additional processing called as capping and telling. कौन undergo करता है? hnRNA undergo additional processing called as capping and telling. In capping, an unusual nucleotide, that is methyl guanosine triphosphate, is added to the 5' end of hnRNA. In telling, adenylate residues, that is 200 to 300, कितने? 200 to 300 are added at 3' end in a template independent manner. चेंज करके यहाँ पे template dependent manner कर सकता है, but correct है template independent manner. Clear? In capping, an unusual nucleotide methyl guanosine triphosphate is added to the 5' end of hnRNA. In telling, adenylate residues, that is 200 to 300 are added to the 3' end in a template independent manner. Clear? It is the fully processed hnRNA now called mRNA. It is the fully processed hnRNA now called mRNA that is transported out of the nucleus for translation. The significance of such complexities is now beginning to be understood. The split gene arrangements represent probably an ancient feature of the genome. The split gene arrangement represent probably an ancient feature of the genome. The presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. The presence of introns is reminiscent of antiquity, and the presence of splicing represents the dominance of RNA world. In the recent times, the understanding of RNA and RNA dependent processes in the living system have assumed more importance. Next is we are going to read genetic code. During replication and transcription, a nucleic acid was copied to form another nucleic acid. As these processes are easy to conceptualize on the basis of complementarity. The process of translation requires transfer of genetic information from a polymer of nucleotide to synthesize a polymer of amino acid. Neither does any complementarity exist between nucleotides and amino acid, nor could any be drawn theoretically. There existed ample evidences to support the notion that changes in the nucleic acid, that is genetic material, were responsible for changes in amino acid in proteins. This led to the preposition of the genetic code that could direct the sequence of amino acid during synthesis of proteins. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the preposition and deciphering of genetic code were most challenging. In a very true sense, it is required involvement of scientists from several disciplines: physicist, organic chemist, biochemist, and genetist. It was George Gamow, a physicist, who argued that since there are only four bases, and if they have to code for 20 amino acids, the code should constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. This was a very bold proposition because a permutations combination of 4⁻3, that is 4 into 4 into 4, would generate 64 codons, generating many more codons than required. Providing proof that the codon was a triplet was a more daunting task. The chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecules with defined combination of bases, that is homopolymer and copolymers. Daunting का हिंदी मतलब होता है निरस्त सहित करने वाला, मीन्स जिसका मीनिंग है मेकिंग यू फील स्लाइटली फ्राइटेंड और वरीड अबाउट योर एबिलिटी टू अचीव समथिंग. ठीक है. Next is Marshall Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. Severe Occhio enzyme, that is polynucleotide phosphorylase, was also helpful in polymerizing RNA with defined sequences in a template-independent manner. Marshall Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. Severe Occhio enzyme, that is polynucleotide phosphorylase, was also helpful in polymerizing RNA with defined sequence in a template-independent manner, that is enzymatic synthesis of RNA. Finally, a checkerboard for genetic code was prepared, which is given in table 5.1. ठीक है. The salient features of genetic code are as follow. First, the codon is triplet. 61 codons code for amino acid and 3 codons do not code for any amino acid. Hence, they function as stop codons. Second point is, some amino acids are coded by more than one codon. Hence, the code is degenerate. That is, some amino acids are coded by more than one codon, hence the code is degenerate. Third is, the codon is read in mRNA in a continuous fashion. The codon is read in mRNA in a continuous fashion. There are no punctuation. Fourth point is, the code is nearly universal. For example, from bacteria to human, UUU would code for phenylalanine, that is phe. Some exception to this rule have been found in mitochondrial codons and in some protozoans. एक्सेप्शन कहाँ फाइंड हुए हैं? माइटोकॉन्ड्रियल कॉडोन्स में एंड इन सम प्रोटोजोन्स में सम एक्सेप्शन फाइंड हुए हैं. ठीक है. फिफ्थ पॉइंट इज AUG हैज ड्यूअल फंक्शन. AUG हैज ड्यूअल फंक्शंस. It codes for methionine that is made and it also acts as initiator codon. कौन सा codon? AUG. ठीक है, ड्यूअल फंक्शन. सिक्स्थ पॉइंट इज UAA, UAG, UGA आर स्टॉप टर्मिनेटर कोडॉन्स. दैट इज UGA, UAA, UAG, UGA. ठीक है, आर स्टॉप टर्मिनेटर कोडॉन्स. कौन कौन? UAA, UAG, UGA. ठीक है. If following is the sequence of nucleotide in mRNA, predict the sequence of amino acid coded by it. Take up help of the checkerboard. AUG दी हुई है, कर सकते हो. ठीक है. Now try the opposite. Following is the sequence of amino acid coded by an mRNA. Predict the nucleotide sequence in the RNA. That is met phi. Do you face any difficulty in predicting the opposite? Can you now correlate which two properties of genetic code you have learnt? Next is mutations and genetic code. The relationship between genes and DNA are best understood by mutation studies. You have studied about mutation and its effect in chapter 4. Effects of large deletion and rearrangement in a segment of a DNA are easy to comprehend. It may result in loss or gain of a gene and so a function. The effect of point mutation will be explained here. A classical example of point mutation is a change of single base pair in the gene for a beta globin chain that results in the change of amino acid residue glutamate to valine. It results in a diseased condition called as sickle cell anemia. इसका एग्जाम्पल है पॉइंट म्यूटेशन का. Effect of point mutation that inserts or deletes a base in a structural gene can be better understood by following simple example. Consider a statement that is made up of the following words that each having 3 letters like genetic code. Ram has red cap. If we insert a letter B in between has and red and rearrange the statement, it would be read as Ram has BDCAP. If similarly we insert the 2 letter at the same place, say BI, now it would read Ram has BID-EDCAP. Now we insert 3 letters together as big. The statement would be read as Ram has big red cap. The same exercise can be repeated by deleting the letter R, E and D one by one and rearranging the statement to make a triplet word. That is Ram EDCAP, Ram has DCAP, Ram has cap. ठीक है. The conclusion from the above exercise is very obvious. Insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. However, such mutations are referred to as frame shift insertion or deletion mutations. insertion or deletion of three or its multiple bases, insert or delete in one or multiple codon, hence one or multiple amino acids and reading frame remains unaltered from that point onwards. Insertion or deletion of three or its multiple base, insert or delete in one or multiple codon, has one or multiple amino acid and reading frame remains unaltered from that point onwards. So the next topic is tRNA, the adapter molecule. From the very beginning of the preposition of code, it was clear to Francis Crick that there has to be a mechanism to read the code and also to link it to the amino acid, because amino acids have no structural specialties to read the code uniquely. He postulated the presence of an adapter molecule that would on one hand read the code and on the other hand would bind to a specific amino acid. The tRNA, then called sRNA, that is soluble RNA, was known before the genetic code was postulated. However, its role as an adapter molecule was assigned much later. tRNA has an anticodon loop. tRNA has an anticodon loop that has bases complementary to the code, and it also has an amino acid acceptor end to which it binds to the amino acids. tRNA are specific for each amino acid. tRNA are specific to each amino acid. For each amino acid. Okay? For initiation, there is another specific tRNA that is referred to as initiator tRNA. There are no tRNA for the stop codon. There are no tRNA for stop codon. In figure 5.12, that is tRNA, the adapter molecule, the secondary structure of tRNA has been depicted that looks like a clover leaf. Secondary structure look like a clover leaf. In actual structure, the tRNA is a compact molecule which looks like inverted L. Okay? Next is translation. Translation refers to the process of polymerization of amino acids to form a polypeptide. The order and sequence of amino acids are defined by the sequence of bases in the mRNA. The amino acids are joined by a bond which is known as peptide bond. Formation of peptide bond requires energy. Therefore, in the first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called as charging of tRNA or amino acylation of tRNA to be more specific. If two such charged tRNA are brought close enough, the formation of peptide bond between them would be favored energetically. The presence of a catalyst would enhance the rate of peptide bond formation. The cellular factory responsible for synthesizing proteins is the ribosome. The ribosome consists of structural RNAs and about 80 different proteins. कितने? 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of translation of the mRNA to protein begins. When the small subunit encounters an mRNA, the process of translation of the mRNA to protein begins. There are two sites in the large subunit. कितनी? There are two sites in the large subunit for subsequent amino acids to bind to and thus be close enough to each other for the formation of a peptide bond. The ribosome also acts as a catalyst that is 23S RNA in bacteria is the enzyme ribosome. कौन सा कैटलिस्ट का रोल प्ले करता है? 23S RNA इन बैक्टीरिया इज द एंजाइम राइबोजाइम. ठीक है. फॉर द फॉर्मेशन ऑफ पेप्टाइड बॉन्ड, a translational unit in mRNA is the sequence of RNA that is flanked by the start codon AUG and the stop codon and codes for polypeptide. A translational unit in mRNA is the sequence of RNA that is flanked by the start codon AUG and the stop codon and codes for polypeptide. An mRNA also has some additional sequences that are not translated and are referred to as untranslated RNAs, that is UTR. The untranslated RNAs UTR are present at both 5' end before the start codon and 3' end and at 3' end after stop codon. Before start codon, that is 5' end, and at 3' end, that is after stop codon. They are required for efficient translation process on UTR. For initiation, the ribosome binds to the mRNA at the start codon AUG, that is recognized only by the initiator tRNA. The ribosome proceeds to the elongation phase of protein synthesis. During this stage, complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with tRNA anticodon. With the tRNA anticodon, the ribosomes moves from codon to codon along the mRNA. Amino acids are added one by one, translated into polypeptide sequence dictated by DNA and represented by mRNA. At the end, a release factor binds to the stop codon. Terminating translation and releasing the complete polypeptide from the ribosome. Next is regulation of gene expression. Regulation of gene expression refers to a very broad term that may occur at various levels. Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be exerted at first transcriptional level, that is formation of primary transcript. Second is processing level, that is regulation of splicing. Third is transport of mRNA from nucleus to the cytoplasm. Fourth is translational level. Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryote, the regulation could be exerted first at transcriptional level, that is formation of primary transcript. Second, that is processing level, that is regulation of splicing. Third, that is transport of mRNA from nucleus to the cytoplasm. Transport of mRNA from the nucleus to the cytoplasm. Fourth is translational level. Clear? The genes in a cell are expressed to perform a particular function or a set of functions. For example, if an enzyme called beta-galactosidase is synthesized by E. coli, it is used to catalyze the hydrolysis of a disaccharide lactose into glucose and galactose. The bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of the enzyme beta-galactosidase. Therefore, in simple terms, it is the metabolic, physiological or environmental conditions that regulate the expression of gene. कौन सी मेटाबॉलिक, फिजियोलॉजिकल और एनवायरमेंटल कंडीशन होती है जो रेगुलेट करती है एक्सप्रेशन ऑफ जीन. द डेवलपमेंट एंड डिफरेंशिएशन ऑफ एम्ब्रियो into adult organisms are also result of the coordinated regulation of expression of several sets of genes. In prokaryotes, control of the rate of transcriptional initiation is the predominant site for control of gene expression. Again I am repeating it. In prokaryotes, control of the rate of transcriptional initiation is the predominant site for control of gene expression. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affect its ability to recognize start sites. These regulatory proteins can act both positively activators and negatively repressors. Positively activators and negatively repressors. The accessibility of promoter lines of prokaryotic DNA is in many cases regulated by interaction of proteins with sequence termed operators. The accessibility of promoter lines of prokaryotic DNA in many cases regulated by the interaction of the protein with sequence termed operators. The operator line is adjacent to the promoter line. The operator line is adjacent to the promoter element in most operons, and in most cases the sequence of the operator binds a repressor protein. Operator binds a repressor protein. Each operon has its specific operator and specific repressor. Clear? For example, leg operator is present only in the leg operon. Leg operator is present only in the leg operon and it interacts specifically with leg repressor only. And it interacts specifically with the leg repressor only. So the next topic is the leg operon. The elucidation of the leg operon was also a result of a close association between a genetist Francis Jacob and a biochemist जैकी मोनाट. जेनेटिस्ट कौन है? फ्रांसिस जैकब, जे से याद रख सकते हो, और बायोकेमिस्ट है जैकी मोनाट, BM से याद रख सकते हो. ये they were the first to elucidate a transcriptionally regulated system. In leg operon, here leg refers to lactose, a polycystonic structural gene is regulated by a common promoter and a regulatory gene. Such arrangement is very common in bacteria and is referred to as operon. To name few such examples: leg operon, trip operon, ara operon, his operon, well operon, etc. The leg operon consists of one regulatory gene, the I gene. Here, the term I does not refer to inducer. Rather, it is derived from the word inhibitor. Does not refer to inducer. Clear? And the three structural genes, that is Z, Y and A. The I gene codes for the repressor of the leg operon. जिसे याद रख सकते हो IREG. The Z gene codes for beta galactosidase, that is beta gal. ZB से याद रख सकते हो, which is primarily responsible for the hydrolysis of the disaccharide lactose into monomeric units, that is glucose and galactose. The Z gene codes for beta galactoside beta gal, which is primarily responsible for the hydrolysis of the disaccharide lactose into monomeric unit galactose and glucose. Clear? The Y gene codes for permease, which increases permeability of the cell to beta galactosidase. The A gene encodes a trans-acetylase. The A gene encodes a trans-acetylase. Hence, all the three gene products in leg operon are required for metabolism of lactose. In most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. Clear? डायग्राम दिया हुआ है यहाँ पे लैक्टोपोरोन का, जहाँ पे प्रमोटर के बाद आई जीन दिया हुआ है, फिर पी दिया हुआ है, फिर ओ दिया हुआ है ZYA in absence of inducer, repressor mRNA. In absence of inducer, repressor binds to the operator ring and prevents RNA polymerase from transcribing the operon. And in presence of inducer, they binds. Okay. So the next, lactose is the substrate for the enzyme beta galactosidase, and it regulates switching on and off of the operon. Hence, it is termed as inducer. In the absence of a preferred carbon source such as glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cell through the action of permease. Remember, a very low level of expression of lac operon has to be present in the cell all the time. Otherwise, lactose cannot enter the cells. Again, I am repeating it. The lactose is transported into the cells through the action of permease. Remember, a very low level of expression of lac operon has to be present in the cell all the time. Otherwise, lactose cannot enter the cell. The lactose then induces the operon in the following manner. The repressor of the operon is synthesized, that is all the time constitutively, from the IG. The repressor of the operon is synthesized from the IG. The repressor protein binds to the operator ring of the operon and prevents RNA polymerase from transcribing the operon. In the presence of an inducer such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. In the presence of an inducer such as lactose or allolactose, the repressor is inactivated by the interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. Essentially, regulation of lac operon can be visualized as regulation of enzyme synthesis by its substrate. Clear? Remember, glucose or galactose cannot act as inducer for lac operon. Glucose or galactose cannot act as inducers for the lac operon. Can you think for how long the lac operon would be expressed in the presence of lactose? I am repeating that paragraph once again. The repressor of the operon is synthesized, that is all the time constitutively, from the IG. The repressor protein binds to the operator ring of the operon and prevents RNA polymerase from transcribing the operon. In the presence of an inducer such as lactose or allolactose, the repressor is inactivated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. Essentially, regulation of lac operon can also be visualized as regulation of enzyme synthesis by its substrate. Remember, glucose or galactose cannot act as inducers for lac operon. कौन नहीं कर सकता? Glucose or galactose cannot act as inducer for the lac operon. Can you think for how long the lac operon would be expressed in the presence of lactose? Regulation of lac operon by repressor is referred to as negative regulation. Lac operon is under the control of positive regulation as well. But it is beyond the scope of discussion at this level. Next topic is Human Genome Project. In the preceding section, you have learned that it is the sequence of bases in the DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequences. If two individuals differ, then their DNA sequence should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques, where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. Human Genome Project was called a mega project. You can imagine the magnitude and the requirement for the project if we simply define the aims of the project is as follow. ह्यूमन जीनोम प्रोजेक्ट कब लॉन्च हुआ था? इन द ईयर 1990. ठीक है, कंफ्यूज करने के लिए 1900 आएगा बट करेक्ट है 1990. So if we simply define the aims of the project as follows. Human genome is said to have approximately 3 into 10 to the power 9 base pair, and if the cost of sequencing required is $3 per base pair, the estimated cost in the beginning, the total estimated cost of the project would be approximately $9 billion. Further, if the obtained sequences were to be stored in the typed form in books, and if each page of the book contained 1000 letters and each book contained 1000 pages, then 1300 such books would be required to store information of DNA sequence from a single human cell. एक ह्यूमन सेल से स्टोरेज करने के लिए इंफॉर्मेशन कितनी बुक चाहिए? 1300 such books. The enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for data storage and retrieval and analysis. Human Genome Project was closely associated with the rapid development of new era in biology called bioinformatics. Human Genome Project was closely associated with the rapid development of a new area in the biology called bioinformatics. Goals of Human Genome Project. Some of the important goals of the Human Genome Project were as follows. First, identify all the approximately 20,000 to 25,000 genes in human DNA. कितने? Identify all the approximately 20,000 to 25,000 genes in the human DNA. 20,000 to 25,000 genes in human DNA. Second point, determine the sequences of the 3 billion chemical base pairs that make up human DNA. Determine the sequences of the 3 billion chemical base pair that make up human DNA. Third is store the information in databases. Fourth is improve tools for data analysis. Fifth is transfer related technologies to other sectors such as industries. Sixth is address the ethical, legal and social issues, that is LC, that may arise from the project. Sixth point है address the ethical, legal and social issues LC that may arise from the project. Fifth point है, again I am repeating that, fifth, transfer related technologies to other sectors such as industries. Next paragraph. The Human Genome Project was 13-year project coordinated by the US Department of Energy and the National Institute of Health. Coordinated by the US Department of Energy and the National Institute of Health. During partner, additional contribution come from came from Japan, France, Germany, China, and others. The project was completed in 2003. चालू कब हुआ था? 1919. Complete कब हुआ है? 2003. The project was completed in 2003. Knowledge about the effect of DNA variation among individuals can be led to the revolutionary new ways to diagnose. Knowledge about the effect of DNA variation among individuals can lead to the evolutionary new ways to diagnose, treat, and someday prevent the thousands of disorders that affect human beings. Besides providing clues to understanding human body, besides providing clues to understanding the human biology, learning about non-human organisms and human organisms can be led to an understanding of their natural capabilities that can be applied towards solving challenges in healthcare, agriculture, energy production, environmental remediation. Many non-human model organisms such as yeast, such as bacteria, yeast, क्या है? Non-human model organism such as bacteria, yeast, Cyanur habitats elegans, that is a free living non-pathogenic nematode जिसका नाम है Cyanur habitatis. Drosophila, the fruit fly, plants, rice, and Arabidopsis, etc. have also been sequenced. Clear? Again, I am repeating that line. Many non-human model organisms such as bacteria, yeast, Cyanur habitats elegans, a free-living non-pathogenic nematode. Drosophila, plants, rice, and Arabidopsis, etc. have been also sequenced. Methodologies. The methodologies involved two major approaches. One approach focused on identifying all the genes that are expressed as RNA, referred to as expressed sequence tags. The other took the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequence, and the latter assigning different ranks in the sequence function, a term referred to as sequence annotation. For sequencing the total DNA from a cell, is isolated and converted into random fragments of relatively smaller size. Recall, DNA is a very long polymer, and there are technical limitations in sequencing very long piece of DNA and cloned in suitable host using specialized vectors. The cloning results into the amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease. So that it can be subsequently sequenced with ease. The common used host were bacteria and yeast, and the vector are called as bacterial artificial chromosome and yeast artificial chromosome, that is bake and yolk, are the two examples of this. The cloning resulted into amplification of each piece of DNA fragment so that subsequently could be sequenced with ease. The common used host were bacteria and yeast, and the vector are called PSC or YSC. Clear? Bacterial artificial chromosome or yeast artificial chromosome. Complete? The fragments were sequenced using automated DNA sequencers that worked on the principle of a method developed by Frederick Sanger. The fragments were sequenced using automated DNA sequencers that worked on the principle of a method developed by Frederick Sanger. Remember, Sanger is also credited for developing methods for determination of amino acid sequence in proteins. This sequence were then arranged based on some overlapping lines present in them. This required generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed only in May 2006. It's an important line. The sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosomes, 22 autosomes and X and Y to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. One line important है, the sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosomes, 22 autosomes and X and Y to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. This was generated using information on polymorphism of restriction endonuclease recognition sites and repetitive DNA sequence known as microsatellites. One of the applications of polymorphism in repetitive DNA sequence shall be explained in a next section of DNA fingerprinting. This was generated using information on polymorphism of restriction endonuclease recognition sites and some repetitive DNA sequences known as microsatellites. One of the applications of polymorphism in repetitive DNA sequence shall be explained in next section of DNA fingerprinting. Next is salient features of human genome. Some of the salient observations drawn from the Human Genome Project are as follows. चलो ध्यान से सुनना, बहुत धीरे धीरे चलेंगे, ठीक है, और अभी यहीं पे क्लियर कर देंगे माइंड में, ठीक है, माइंड में सब सेट कर देंगे अभी. फर्स्ट पॉइंट है, द ह्यूमन जीनोम कंटेन्स 3164.7 million base pair. The human genome contains 3164.7 million base pair. कितने? 31. कितने होते हैं? 4. लास्ट में गया उसके पहले 6, ऐसे मत याद रखो, ठीक है. 3164.7 million base pair. Clear? Second point is, the average gene consists of 3000 bases, but sizes vary greatly, with the largest human gene being dystrophin at 2.4 million bases. Largest gene कौन सा है? Human gene being dystrophin at 2.4 million bases. Again I am repeating it. The average gene consists of 3000 bases, but sizes vary greatly, with the largest non-human gene being dystrophin at 2.4 million bases. Third point is, the total number of genes is estimated at 30,000, much lower than the previous estimates of 80,000 to 1,40,000 genes. Almost all, that is 99.9% nucleotide bases are exactly same in all people. कितने? 99.9% nucleotide bases are exactly same in all people. Fourth is, the function are unknown for over 50% of the discovered gene. Changes करके कुछ भी कर सकता है यहाँ पे, but correct है. The function are unknown for over 50% of the discovered gene. Fifth point, less than 2% of the genome codes for protein. Less than 2% of the genome codes for proteins. Sixth is, repetitive sequences make up very large portion of the human genome. Repetitive sequences make up very large portion of the human genome. Seventh point is, repetitive sequences are stretches of DNA sequence that are repeated many times, sometimes hundreds to 1,000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Eighth point is chromosome 1 has most gene, that is 2968. Chromosome 1 has most gene, that is 2968, and the Y has the fewest, that is 231. ऐसे याद रख सकते हो, लास्ट में 1, ठीक है, फिर फर्स्ट से चालू होगा 2 3, मीन्स 2 3 और 1 सबसे लास्ट में, ठीक है. 1 has most gene 2968 and Y has fewest, that is 231, 231. Ninth point is scientists have identified about 1.4 million locations where single base differences, that is single nucleotide polymorphism, pronounced as SNPs, full form SNPs, occurs in humans. This information promises to revolutionize the process of finding chromosomal location for disease-associated sequences and tracing human history. Scientists have identified about 1.4 million locations. कितनी 1.4 million locations where single base DNA, that is SNPs, occurs in human. SNP का full form है single nucleotide polymorphism. This information promises to revolutionize the process of finding chromosomal locations for disease-associated sequences and tracing human history. Next is application and the future challenges. Deriving meaningful knowledge from the DNA sequences will define research through the coming decades, leading to our understanding of biological systems. This enormous task will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both the public and private worldwide. From varied disciplines in both the public and the private sectors worldwide. One of the greatest impacts of having the human genome sequence may well be enabling a radically new approach to biological research. In the past, researchers studied one or a few genes at a time with whole genome sequences and new high throughput technologies. We can approach questions systematically and on a much broader scale. They can study all the genes in a genome, for example, all the transcripts in a particular tissue or organs or tumor, or how tens of thousands of genes and proteins work together in interconnected networks to orchestrate the chemistry of life. Next is DNA fingerprinting. As stated in the preceding section, that is 99.9% of base sequence among humans is the same. Assuming human genome as 3 into 10 to the power 9 base pair, in how many base sequences would there be differences? It is these differences in sequence of DNA which make every individual unique in their phenotypic appearance, which make every individual unique in their phenotypic appearance. Clear? If one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be a daunting and expensive task. Imagine trying to compare two sets of 3 into 10 to the power 6 base pair. DNA fingerprinting is a very quick way to compare the DNA sequences of any two individuals. DNA fingerprinting involves identifying differences in some specific lines in DNA sequence called as repetitive DNA. DNA fingerprinting involves identifying differences in some specific lines in DNA sequence called as repetitive DNA, because in these sequences a small stretch of DNA is repeated many times. This repetitive DNA are separated from bulk genomic DNA as a different peak during density gradient centrifugation. The bulk DNA forms a major peak and the other peak are referred to as satellite DNA. The bulk DNA forms a major peak and the other small peaks are referred to as satellite DNA. Depending on base composition, that is AT reach or GC reach, length of segment and number of repetitive units, a satellite DNA is classified into many categories such as microsatellites, mini satellites, etc. इस पे depending on base composition A is to T reach or G is to C reach, length of segment and number of repetitive units. The satellite DNA is classified into many categories such as microsatellite and mini satellites, etc. These sequences normally do not code for any proteins, but they form a large portion of human genome. This sequence show high degree of polymorphism and form the basis of DNA fingerprinting. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue such as blood, hair follicle, skin, bone, saliva, sperm, etc. from any individual show the same degree of polymorphism, they become very useful identification tool in forensic application. ठीक है? Understood? Clear? Further, as the polymorphisms are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting, it is essential that we understand that what DNA polymorphism means in simple terms. ये क्वेश्चन पूछा हुआ है, ठीक है, PYQ. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting. क्या है? Polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting. It is essential that we understand what DNA polymorphism means in simple terms. Polymorphism, that is variation at genetic level. Variation at genetic level arises due to mutation. Recall different kinds of mutations and their effect that you have already studied in chapter 4 and in the preceding section in this chapter. New mutations may arise in an individual either in somatic cells or in the germ cells. Germ cells are the cells that generate gametes in sexually reproducing organisms. If a germ cell mutation does not seriously impair individual's ability to have offspring who can transmit the mutation, it can spread to the other members of population through sexual reproduction. Allelic. Again, recall the definition of alleles from chapter 4. Allelic sequence variation has traditionally been described as a DNA polymorphism if more than one variant, that is allele at a locus, occurs in human population with a frequency greater than 0.01. ठीक है, allelic sequence variation has traditionally been described as a DNA polymorphism if more than one variant, that is allele at a locus, occurs in human population with a frequency greater than 0.01. यहाँ से क्वेश्चन आते ही आता है DNA फिंगरप्रिंटिंग से, पक्का शॉर्ट क्वेश्चन आता है यहाँ से. So let's proceed further. In simple terms, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. Again, I am repeating it. In simple terms, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. The probability of such variation to be observed in non-coding DNA sequences would be higher as mutation in these sequences may not have any immediate impact in an individual's reproductive ability. Clear? The probability of such variation to be observed in non-coding DNA sequence would be higher, as mutation in this sequence may not have an immediate impact in an individual's reproductive ability. ठीक है, कहाँ पे ज़्यादा होगा? probability of such variation to be observed in non-coding DNA sequence would be higher, because as mutation in this sequence may not have an immediate impact in an individual's reproductive ability. Capability नहीं, ability. Clear? These mutations keep on accumulating generation after generation, and from one of the bases of variability, polymorphism. There is a variety of different types of polymorphism ranging from single nucleotide change to a very large scale changes. For evolution and speciation, such polymorphism play a very important role. For evolution and speciation, such polymorphism play a very important role, and you will study these in details at higher class. The technique of DNA fingerprinting was initially developed by Alec Jeffreys. He used a satellite DNA as a probe that show very high degree of polymorphism. कौन शो करता है? Probe. He used a satellite DNA as probe that shows very high degree of polymorphism. It was called as variable number of tandem repeats, VNTR. कौन? Probes. ठीक है. It was called as variable number of tandem repeats, that is VNTR. The technique as used earlier involved southern blot hybridization using radio-labeled VNTR as a probe. It included first, that is isolation of DNA, second, digestion of DNA by restriction endonuclease, third, separation of DNA fragments by electrophoresis, fourth, transferring blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon, fifth, hybridization using labeled VNTR probe, and sixth, detection of hybridized DNA fragments by autoradiography. A schematic representation of DNA fingerprinting is shown in the figure 5.60. एक बार और पॉइंट्स रिपीट कर रहा हूँ. फर्स्ट पॉइंट है आइसोलेशन ऑफ डीएनए, सेकंड डाइजेशन ऑफ डीएनए बाय रेस्ट्रिक्शन एंडोन्यूक्लियस, थर्ड separation of DNA fragments by electrophoresis, fourth transferring blotting of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon. ठीक है. फोर्थ पॉइंट है ट्रांसफर ऑफ separated DNA fragments to synthetic membrane such as nitrocellulose or nylon. Fifth, hybridization using labeled VNTR probe. And sixth, detection of hybridized DNA fragment by autoradiography. ठीक है. लास्ट पॉइंट है डिटेक्शन ऑफ हाइब्रिडाइज्ड DNA fragment by autoradiography. So let's proceed further. The VNTR belongs to a class of satellite DNA, refers to as mini satellite. VNTR कौन से क्लास से बिलोंग करते हैं? मिनी सैटेलाइट. ठीक है. The VNTR belong to a class of satellite DNA refers to as mini satellite. A small DNA sequence is arranged tandemly in many copy numbers. The copy number varies from chromosome to chromosome in an individual. The number of repeats show very high degree of polymorphism. As a result, the size of VNTR, that is variable number of tandem repeats, varies in size from 0.1 to 20 kb. ये important line है. As a result, the size of VNTR varies in size from 0.1 to 20 kb. Consequently, after hybridization with VNTR probe, the autorradiogram gives many bands of different sizes. These bands give a characteristic pattern for an individual DNA. ठीक है. Consequently, after hybridization with VNTR probe, the autoradiogram gives many bands of differing sizes. These bands give a characteristic pattern for an individual DNA. It refers from individual to individual in a population, except in the case of monozygotic identical twins. It differs from individual to individual in a population except in the case of monozygotic identical twins. The sensitivity of the technique has been increased by use of polymerase chain reaction, that is PCR. You will study about it in chapter 9. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to the application in forensic science, it has much wider application such as in determining population and genetic diversity, such as determination and the determining population and genetic diversities. Currently, many different probes are used to generate DNA fingerprints. डायग्राम दिया हुआ है यहाँ पर, ठीक है. Schematic representation of DNA fingerprinting. Few representative chromosomes have been shown to contain different copy number of VNTR. For the sake of understanding, different color schemes have been used to trace the origin of each bands in the gel. The two alleles, that is paternal and maternal of a chromosome, also contain different copy number of VNTR. It is clear that the binding pattern of the DNA from crime scene matches with the individual B and not with A. Now we are going to read the summary. Nucleic acid are the long polymers of nucleotides. While DNA stores genetic information, RNA mostly helps in transfer and expression of information. DNA store करेगा genetic information. RNA mostly helps in the transfer and expression of information. तो DNA and RNA both function as the genetic material, but DNA being chemically and structurally more stable is a better genetic material. However, RNA is the first to evolve and DNA was derived from RNA. The hallmark of the double-stranded helical structure of DNA is the hydrogen bonding between the bases from opposite strands. The rule is that adenine pairs with thymine through 2 hydrogen bonds and guanine with cytosine through 3 hydrogen bonds. This makes one strand complementary to the other. The DNA replicates semi-conservatively. The process is guided by the complementary hydrogen bonding. A segment of DNA that codes for RNA may, in a simplistic term, can be referred to as gene. During transcription also, one of the strands of DNA acts as template to direct the synthesis of complementary RNA. In bacteria, the transcribed mRNA is functional, hence can directly be translated. In eukaryotes, the gene is split. In eukaryotes, the gene is split. The coding sequences exons are interrupted by non-coding sequences. Exons are interrupted by non-coding sequences, that is introns. Introns are removed and exons are joined to produce functional RNA by splicing. The messenger RNA contains the base sequences that are read in a combination of 3 to make triplet genetic code to code for an amino acid. The genetic code is read again on the principle of complementarity by tRNA that acts as an adapter molecule. There are specific tRNAs for every amino acid. The tRNA binds to a specific amino acid at one end and pairs through hydrogen bonding with codes on mRNA through its anticodons. The site of translation, that is protein synthesis, is ribosomes. Site of translation is ribosomes, which bind to mRNA and provide platform for joining of amino acids. One of the rRNA acts as a catalyst for peptide bond formation, which is an example of RNA enzyme, that is ribozyme. Translation is a process that has evolved around RNA. In indicating that life began around RNA. Since transcription and translation are energetically very expensive processes, these have to be tightly regulated. Regulation of transcription is the primary step for regulation of gene expression. In bacteria, more than one gene is arranged together and regulated in units called as operons. Leg operon is the prototype operon in bacteria, which codes for genes responsible for metabolism of lactose. The operon is regulated by the amount of lactose in the medium where the bacteria are grown. Therefore, this regulation can also be viewed as regulation of enzyme synthesis by its substrate. Human Genome Project was a mega project that aimed to sequence every base in human genome. This project has yielded much new information. Many new areas and avenues have opened up as a consequence of project. DNA fingerprinting is a technique to find out variations in individuals of a population at DNA level. It works on the principle of polymorphism in DNA sequences. It works on the principle of polymorphism in DNA sequences. It has immense application in the field of forensic science, genetic biodiversity, and evolutionary biology. कौन सी फील्ड में? forensic science, genetic biodiversity, and evolutionary biology. With this, we completed the chapter. Thank you.